So will anybody be kind enough to tell me what is an RNA? Ribonucleic acid. Okay. What do you mean by the term? हम क्लोरमेंट क्यों खाते हैं? When you are saying or using the term ribonucleic acid, what do you mean? Come on, tell me. Yes, very good. It's a nucleic acid that is either a purine or a pyrimidine with a ribose sugar. Okay, with a ribose sugar. What kind of a sugar is ribose sugar? It's a pentose sugar. Very good. Okay. So it can be reduced at 2 points. If there is only 1 point reduction, then it is ribonucleic acid. When it is reduced at 2 points, then it is deoxyribonucleic acid. Okay. Can anyone tell me what are the names of purines and pyrimidines? Very good. So I usually remember it via the Chargaff's rule. Okay, ATGC. Okay. What is the special difference between DNA and RNA in the purine pyrimidine base pairing? This is basic 12. Thymine is replaced by uracil. Very good. Thymine is replaced by uracil. So RNAs do have a broad classification. Okay. And the broad classification is they are either coding RNAs or they are non-coding RNAs. Can anyone tell me what are they? What's a coding RNA? What's a non-coding RNA? Yes, very good. Coding RNA contributes in protein primary structure generation. Okay, protein primary structure generation. That is, these are mostly strands which will be utilized for what? Transcription or translation? Translation. Okay, translation. That is only 20% of the RNA which is present. So do you think rest of the RNA is junk? Is it junk? Absolutely not. So with coding RNAs, you have non-coding RNAs. Now non-coding RNAs can be of two types. Okay, regulatory RNAs and non-regulatory RNAs. Regulatory RNAs and non-regulatory RNAs. What is meant by regulatory RNAs is the RNAs that play an important role in protein formation. They are not the structural components but the regulatory components. And non-regulatory RNAs are structural RNAs. They present you the scaffolding or the support system in which RNA is being produced or RNA expression is being modulated. When I am using the term RNA expression, what I am trying to mean? DNA expression, RNA expression, what I am trying to mean? The amount of translations that an RNA will undergo for formation of proteins. That is your RNA expression. So the entire role of genetic expression will depend upon the RNA world. It will provide the bench it will provide the informatics, it will regulate the rate, and it will manage differential expression. Differential expression. Right? You have a beautiful cornea. Okay? From one cell, single cell, that cornea was produced. From that single cell, your neuron was also produced. How did that happen? Selective expression. Selective expression of DNA from a single totipotent or multipotent stem cell. The entire body came into being. So what will become what and how much of it will function is totally modulated at the stage of RNA. Are we clear on that? Any doubts till now? So RNA is a polymer. RNA is a polymer, right? And this polymer is being held together by what kind of a linkage? Phosphodiester linkage. Okay. It consists of a phosphodiester linkage. It starts at the nucleolus and finishes in the cytoplasm. This entire expression is regulated at the base of RNA. Whether a stem cell will become an RBC, whether it will become a muscle, whether it will become an osteoblast, everything will be decided at this level via genetic expression. Also, once they have taken into their proper tissue shape, the amount of proteins a single muscle cell will have, that is also being regulated at this level. Okay, clear till now? So let us look at the functions of RNA from a holistic approach. Now we have got the major points, right? First of all is protein coding. The first function and the most primary function is protein coding. Second function is very good transfer of genetic material occurs via DNA from one species, from one unit to species to the second unit. But here what you are trying to say or what you are trying to mean is the genetic expression is being regulated at tissue level. Okay, so whenever you state an answer that needs to carry particular catchwords which will indicate towards the right answer. So you have to be beware, and you also have to be used to regulated speech so that you do not throw a confusing picture to your examiner. So coding RNA was mRNA. Then you have got non-coding RNA, that is either regulatory RNA or non-regulatory RNA. Non-regulatory RNAs are what? Structural RNAs. So under this classification, in your coding RNA we will have mRNAs. In coding RNAs we will have mRNAs. Okay. And in non-coding RNAs, in non-regulatory RNAs, we will have the two major classes, and they are ribosomal RNA or rRNA and your tRNA. Ribosomal RNA and your tRNA. tRNA also known as transfer RNA. And then you have regulatory RNAs. Now that is going to be a long list. Okay. First of all, there is siRNA, then there is miRNA, then there is piRNA, then there are short nuclear segments. Okay. So in that class we have about 5 to 8 subclasses of regulatory RNAs. Okay. So messenger RNA. How does it look like? It has got a 5 prime cap. Then it has got only histonic segments. Okay, there is a UTR region, then there is your histonic segments. At the end of histonic segments, there is another 3 prime UTR region followed by a polyadenylated A or poly AA. For those who are wondering how it looks like this. Okay, this is the 5 prime methyl guanosine element. It serves a protective function because in your cell, in your cytoplasm, and even in your nucleus, you have RNase enzymes. You have RNase enzymes, and human RNAs are deterred from digesting human origin mRNAs by the presence of this methyl guanosine element, and that is how you have the ability to destroy the RNAs that are created from bacterial viruses. So it also plays a profound role in immunity, different autoimmune disorders, and definitely a huge role in cancer genetics. After this 5 prime methyl guanosine cap, you have a UTR. What is the use of this UTR region? Very good. It is untranslated. So why it is there? Why nature did seem fit? No, it is the binding place of your RNA. It is the binding place for your RNA polymerase. And the amount of binding that will take place and the amount of cycles of translation that will take place is also dependent upon this UTR region, which is present at the 5 prime end. As I said, nice, lovely line faces. Okay, but there are 2 UTR regions. At the 5 prime UTR region, then you have the cystonic segment. Cystonic segment everybody knows. Got taught in 10, 12 sufficient. What about the later UTR region? So whatever has a beginning has an end. So if an mRNA is produced, it will also be digested. So when it will be digested is determined by the later 3 prime UTR end. It is said if the polyadenylated tail grows smaller than 12 adenosine units, then this mRNA will be digested. Okay. So what is this poly-A tail doing? It is providing protection from cellular and nuclear RNAs. Okay, it is protecting from cellular and nuclear RNAs. So that is the main things about your mRNA. See, 5 prime cap, 5 prime UTR, coding sequence, 3 prime UTR, poly A tail. That is the structure from 5 prime end to your 3 prime end. Now we come to tRNA, clover leaf model. Okay, clover leaf model. Here you see RNAs have the ability to bend upon each other, and create loops and bends. Why is tRNA so important? It recognizes the triplet codon. In your cystonic segments. So obviously all proteins will be starting with AUG, which is a triplet codon, and that will be synthesizing with her. Okay. So how does it take place? This fella has got 4 arms. Okay, the tRNA structure has got 4 arms. First is the anticodon arm. The anticodon arm is going to sit on your mRNA, and that is they have just the complementary base pairing. They have the complementary and inverse base pairing. That is the function of your which arm? Anticodon arm. Then you have a D arm. You have a T arm and a small variable junction. The most important of them being is the 3 prime acceptor arm. The most important of them being the 3 prime acceptor arm. Because this acceptor arm will be getting hold of the amino acid, the specific amino acid. Okay. Usually D arm is supportive, and the T arm has enzymatic activity. Along with peptidyl transferase, it has got another enzyme activity which fuses one amino acid to the other amino acid to form what kind of a linkage? What kind of linkage does proteins have? Shiva Das man, let her answer. Peptide linkage CONH. Okay, look at peptide linkage CONH. Now these structures of tRNA have some kind of specialized or modified bases. For an example, it has pseudouridine, which is a modified base from uracil. Since I have much to cover, I do not have the time to go into deeper, to go into the structural nuances. If you are lucky, then you have another class on it. If you are not lucky, then you will have to read it up yourself. But it's a very common question. The clover leaf model will appear in your university examination in one way or other. You cannot get rid of it. Okay. This is a high value exam question, and this is a fantastic place to give Nice answers on Viva. So give it a thorough read. Give it a thorough read. Now, rRNAs. In eukaryotes, we have ribosomes, 50S and 30S. What do you mean by S? Sweat work unit. Very good. What do you mean by sweat work unit? Very good. It is the sedimental coefficient or it is the rotational coefficient. The amount of rotational power will cause this particular sedimentation to occur at a particular speed of rotation. Okay, that is what is meant by sweat work units. Right? So, RNA, 50S RNA, 30S RNA. They are also consisted, the entire ribosomal structure will also be consist of several kinds of structural proteins along with your ribosomal RNA. What are they actually making? They are basically making an anvil and a hammer. They are basically making an anvil and a hammer. One is the support system or the support bed in which your mRNA is being placed. On top of that mRNA, you have tRNA, and the hammer being the 30S unit, which has the enzyme activity of peptidyl transferase, so that it can hammer all units together to form a polypeptide chain. Correct? So now, tell me one example of non-protein enzyme. And I just told you, man, ribozyme and peptidyl transferase. Okay, peptidyl transferase and ribozyme. These are the commonest example of non-protein enzymes. Very common question asked in Viva. Teachers love to ask this question. Okay, non-protein enzymes. Apart from that, the rRNAs will be giving the entire support system for translation to happen. Okay, and they also have other enzymatic activities like endonucleases, exonucleases, ATPases, helicases, and leachases. They have so much enzymatic activity, all for the sake of gene expression. All for the sake of gene expression. Now, miRNAs or microRNAs, usually 19 to 25 nucleotides in length. It is a regulatory RNA. It was seen in Caenorhabdis elegans. The miRNA played a special role in the diversification of pluripotent stem cells into totipotent stem cells. Again, big words. God knows what they mean. Pluripotent stem cell, differentiated into totipotent stem cell. But what is this thing? Diversification of pluripotent stem cells into totipotent stem cells. In order to understand this, you have to know what is pluripotent, what is totipotent. So what is pluripotent? Yes, all types of germ lines can be accessed via pluripotent stem cells. totipotent stem cells can also give rise to different tissues of a particular organ system. For example, a pluripotent stem cell can get differentiated to form bone structures, or it can be differentiated to form muscle structures. But totipotent stem cells, they will only give rise to muscle structures, different types of muscle structures, the fibroblasts, the elastin filaments, the coverings and all that, but it cannot give rise to an osteoblast. Got it? Did everybody get the fact? Okay? Pluripotent stem cells can get differentiated into any tissue system. It can get differentiated into tissue systems of the eye. It can get differentiated into systems of your ear, inner ear. It can become bone. It can become cells of cartilage. But once it becomes totipotent, it can only give rise to a single organ structure. If it got differentiated into bone, then it will be able to give rise to osteoblasts, to osteoclasts, to osteofibers, but not corneal structures. Are we clear on that? So this mRNA assures the proper transgression of pluripotent stem cells into totipotent stem cells. Otherwise, had they retained their pluripotency, it would have become cancerous. Okay. So here, mRNAs have a profound impact right from metabolism to immunity. It was discovered right in Clinolabdis, but it is also present in human body. Okay, and they are related into cancer oncotics, cardiovascular diseases, and also metabolic diseases. Okay. See, this is how mRNA will be regulated. This is your mRNA. This is your UTR segment. It might get over here. It might decide to degrade the entire mRNA. If the mRNA gets degraded, then the expression slows down. Similarly, it might sit over here and it might block the protein translation, thereby causing a temporal shift in the protein expression. It is just blocking the RNA. The mRNA is not being degraded. If need arises, this mRNA will release, this myRNA will release the mRNA, and then again translation can occur in the same RNA for expression. Got it? One is a temporal block and one is total degradation. So when an RNA will be degraded, that is being decided, or when it will be blocked from expressing its content, that will be decided by your regulatory values. Another Nobel Prize. Jumping genes. Barbara McClintock. BCR-ABL gene. Jumping genes are also called as transposons. What happens is, a particular sector of your DNA, be it coding or be it non-coding, it will form a hairpin loop. It will break up and then it will re-emerge itself in another location of the gene. The BCR-ABL is the 18th chromosome of BCR-ABL, and on your 20 there is ABL. So here two genes, right? There is a jump of a particular amount of nucleotides from your chromosome 18 to your chromosome 22, forming your BCR-ABL gene complex. And this BCR-ABL gene complex will be continuously translated by tyrosine kinases to produce chronic myeloid leukemia. What these siRNAs do? Whenever there is a jumping action of any transporter, this siRNA is going to clip it, is going to destroy that jump, thereby maintaining your integrity of DNA. In case of this mechanism is lost, you find different diseases like Parkinson's, like Alzheimer's, like multiple sclerosis, so much more, and different types of cancers. Particularly, siRNAs are infused with destroying hairpin loops of genetic materials of viruses, especially that of Epstein-Barr virus. All viruses that have a propensity of causing leukemia, different types of leukemia, siRNA is the most important buffer to tell. RNAs, PV integrated RNAs. They are associated with carbonate proteins. They are associated with carbonate proteins. PiRNAs regulate the amount of translation that will be happening on an mRNA. It is a regulatory protein. And one of the most important roles being in many organisms, piRNAs are essential for fertility and germ cell development. Fertility defects in mutants lacking piRNA can occur in absence of defective T silencing. Silencing means, the word silencing means the stoppage of expression from an mRNA by a regulatory RNA. Okay, so it plays a highly protective action in our gonads, ovaries, testicles, so that the germ cells being produced are protected from mutations. Okay, we have heard of germ-like tumors. You will hear in your unit, they are called gonadal tumors like dysgerminoma, seminoma. These cancers occur when there is a defective piRNA silencing action. Okay, very good. This was an offset program of Human Genome Project. This was an offset program of Human Genome Project. This is a man-made RNA. This is a man-made RNA. So it is not possible to preserve the entire genome of a human being in different gene libraries. Why? Because we do not have any option to replicate the non-coding segments. We can only replicate the coding segments. So what did we do? We got hold of the entire mRNA profile, the entire mRNA profile of a particular cell. Then we use the reverse transcriptase enzyme on it and we made cDNA replicates, and then we store that cDNA replicates in your human genome RNA library. Why? Why did we do it? Suppose this fellow over here, he owns a multi-billion-dollar company. Now he wants to go into pharmaceutical segment. He wants to create and sell human albumin. He wants to sell human albumin. No, even better than that, human erythropoietin. So how will he get hold of this human erythropoietin? He has to start somewhere, no? So he has to go to the gene library, sorry, not the gene library, the mRNA library, and he has to say that please provide me the cDNA for erythropoietin, and they will give it to him in lieu of the cost. He will take it and then he will create his molecular biology lab. Now what will he do? He will convert that cDNA into RNA. He will put it inside a plasmid and he will impregnate that plasmid containing human erythropoietin where? In HeLa cells. HeLa cells are the platform of expression. And then he will utilize those HeLa cells to grow cultures of HeLa cells which have the specific ability to create erythropoietin. Then he will crush those cells and then with the help of chromatography he will separate that erythropoietin protein, and then he is going to convert it into an amorphous powder and he is going to sell it. Right? Where did it all start? Single cell RNA. It all started from single cell RNA. Think, a patient of liver failure, a patient of suppose severe degenerative osteoarthritis. We are using collagen, we are using albumin, we are using thyroid hormones. How? Everything is a product of genetic modification. Maximum replacements, maximum body replacements are a product of that, and that has at least increased the longevity of human beings by 40 years, and that is the impact of genetics and molecular techniques on today's society, though we do not realize it. We only realize it when we go to that industry. Okay, so circular DNA, single stranded. Always remember, circular DNA is single stranded. Do not confuse circular DNA with cDNA. Circular RNA with cDNA, they are absolutely two different things. Okay? What do these circular RNAs do? They are basically found in lower RP. Okay? They are basically found in lower RP. It presents various, lower RP means bacteria and viruses. Okay, that is what I mean by lower RP. So they are not found in eukaryotes. They are not found in eukaryotes. And their analysis is still going on. We do not have much information. We only know that this species is there and it may play a vital function. Okay. Lower RP. SNORNA or small nuclear RNAs. there are two main classes of SNO-RNAs. First is box CD SNO-RNAs and box H ACA SNO-RNAs. These two differ in their structural elements, binding proteins and specific conserved proteins. They are highly conserved because they play the regulatory role in your UTR regions of mRNA. What do you mean by highly conserved? The same set of SNO-RNAs is being preserved in species line. So DNA is not the only molecule which gives us heredity. RNA also plays the role of heredity, but not the majority. But it also does have a role, and that is why SNO-RNAs are preserved in genetic lines, so that your proper tissues in proper time can act in the proper way for you to grow from a single cell into an entire organism. It allows you to go through the ages of infancy, to childhood, to a young adult, to an adult, up till to an old man. This entire age sequencing is being modulated at your RNA and DNA levels. This poly A tail, this is one of the crux property of aging. Your DNA also have your poly A tail structures, and this protects you from the intrinsic DNases that is present in your entire genome. And there is an enzyme which increases the length of this poly A tail. We can utilize that enzyme to ensure that your genetic integrity or the RNA integrity remains for a longer period, thereby allowing us to remain younger for longer periods and age relatively slower. Okay, this enzyme is called telomerase enzyme. This enzyme is called telomerase enzyme, the crux of aging. Okay. Finally, CRISPR, an adaptive immune system. There are two types of immunity in our body. Two types of immunity. Acquired and retained. Inherited. Okay. Innate immunity and acquired immunity. Innate immunity and acquired immunity. See how important this fellow is, CRISPR. What does it do? What does it do? It has specialized Cas proteins. It has specialized Cas proteins which destroy viral and bacterial DNA materials and RNA materials, thereby preventing their integration in the human genomics, preventing diseases and cancers. Preventing diseases and cancers. Which destroys bacterial and viral DNA and RNA, thereby preventing their integration in the human cellular system. Therein saving us from an active infection. Think about retroviruses. HIV is not the single retrovirus in the world. There are other retroviruses as well. This CRISPR mechanism is an RNA mediated mechanism which infers you immunity. Another huge role. See, our body's immunity is active easily with bacteria. Why? Because we can engulf bacteria and phagocytes, we can destroy it easily. Bacteria are relatively larger, but viruses are very small. They can get integrated very easily. That is why in case of viremia, there is a very quick onset of symptoms. In viral diseases, you do not see a convalescent period that is going on 7 days, 14 days incubation period going on. No, in 3 days, done job. Why? It can penetrate very easily. And the only way for a virus to get destroyed is that this virus is digested inside the cell and the viral marker proteins appear in the cell membrane, thereby acting toll-like receptors so that they can be started upon apoptosis. So this entire equation of destroying that viral RNA and to use it as a molecular signal to start the action of innate immunity is totally being provided by your RNAs. Okay? What most students have a tendency to do whenever they are asked about types of RNAs, they only say mRNA, tRNA, rRNA, done. All other RNAs which have a much larger role are usually forgotten and shoved aside. Moral of the story is, if you are asked about the types, then name all of them along with their classification. Okay, name all of them along with their classification. Otherwise, out of 10 points of classification, you are mentioning only 3. And then you come, sir, I only got 2 out of 10. You only mentioned 3 points. Yeah, obviously you are going to get 2. Okay. So with this, we will finish our lecture on types of RNAs. Thank you for your patience.
We are dealing with some infectious agent if in our body enter, right? If any patients come with your fever, respiratory distress, and there is a definite history of fever. Fever means some sorts of infection is there. We are doing the CBC, and there the total leukocyte counts, differential counts are seen in the complete blood count level, right? So, what do you mean by diabetes? People, you. দেখি কিরকম বই খুলছিস টিস্যুগুলো একটু একটু পড়েছিস ওকে এনিথিং আর কেউ কিছু বলবি so neutrophil enter into the site when there is any offending agents and also it invites others soldiers like macrophages, like other friends there of like neutrophils, and in this way they are damages the microbes. Now question is that how? Question is that how? Suppose this is a neutrophil, since the lobes are there and some granules are there. কথা কিন্তু এখনো হচ্ছে আমি কিন্তু দেখলে বের করে দেব so these are the granules all over the neutrophils. So already know the granules are the very much releases some enzymes like দু একটা নাম myeloperoxidase, lactoferrin, defensin, elastase, cathepsin G, আর gelatinase, very good. So these are the inside the granules. Now suppose this is a bacteria. এটা ব্যাকটেরিয়া আছে, একে ইনগালভ করবে, will destroy. Question is that how? There are two mechanisms mainly. One is phagocytosis, another is respiratory burst বা oxidative burst. তাহলে কি বললাম? By the phagocytosis, and another is a respiratory burst, burst. There is another new mechanism that is recently discovered, that is called netosis, by forming neutrophil extracellular traps. Neutrophil extracellular traps. মাথাটা আগে একটু ঠান্ডা কর, নইলে কিছু বুঝতে পারবি না কি সব বলছি। Traps, neutrophil extracellular traps. This is very new. আমি এখানে ডিটেলসে বলতে গেলে 4টে ক্লাস লাগবে, দরকার নেই, আমি এখানে ছোট্ট করে বলে দেবো। Phagocytosis and respiratory burst is the main. Now question is when it is required. সবসময় লাগছে? Suppose there are 10 bacteria are there. Bacteria may be fungal, because remember always in neutrophil not only kill the bacteria, it also kills the fungus. ভাইরাসের জন্য আলাদা cellular soldiers there is there. So suppose this is the bacteria and these are 10 bacteria আছে, and then suppose this is phagosome হচ্ছে। phagosome মানে কি? এখান থেকে কি করবে জিনিসটা, এরকম করে একটা করে ব্যাকটেরিয়াটাকে আমি এখানে এনগালভ করব, তাই তো? নিয়ে আমি একটা ফ্যাগোজোম তৈরি করব। ফ্যাগোজোম উইল ফর্ম এ ফ্যাগোজোম। তাহলে ফ্যাগোজোমটা কি কবে হবে? পুরোটা কভার হয়ে যাবে। আর এখানে কি আছে আমাদের? লাইসোজোম চলে গেল, এখানে একটা ফ্যাগোজোম তৈরি করে এর ভিতরে ঢুকে গেল। লাইসোজোমে কি আছে? লাইসোজাইমস, এনজাইমস। পিএইচ কমিয়ে দিল, ডিক্রিজ দ্য পিএইচ, অ্যান্ড দেন দ্য lysis of the bacteria damage, and then it is called endocytosis, and then this exocytosis will be there, the products of the bacteria will be gone out only for the limited bacteria. এ খেয়েই যাচ্ছে, he is engulfing more and more bacteria, আসছে প্রচুর ব্যাকটেরিয়া আছে, গিলে বসে আছে। So these are the basic problems, more and more ও পাচ্ছে, ও নিচ্ছে, তাই তো? So these are the engulf in the much more, then the phagolysosome will not work properly, or they does not kill properly with the bacterial cidal effect. Then the process of oxidative burst will come. Oxidative burst means formation of some reactive oxygen species. প্রত্যেকটা শর্ট নোট দেয়, reactive oxygen species, reactive oxygen species. Okay, so in this way oxidative formation হবে। কিভাবে হবে এইবারেতে আসবো। Suppose this is a neutrophil and this is a invaginations of the bacteria. Bacteria এসে এখানে বসে আছে। Bacteria, কথা বলছিস এখনো? Bacteria is there, right? Now we are always taking the oxidative burst, respiratory burst. That means some from involvement of the oxygen is there, right? So oxygen. নিচ্ছি আমরা oxygen always. In our blood also oxygen, circulation in our oxygen. So there is no scarcity of any oxygen at least. So when the multiple bacteria engulf or bacterial load is very high, then the oxidative burst as well as netosis by forming the neutrophil extracellular traps will come into force. তারা তখন আসবে। কীভাবে আসবে? তাহলে আমি কি বললাম না, আমাকে রিঅ্যাক্টিভ এয়ারওয়ে ডিজিজ এই স্পিসিস বানিয়ে একে মারতে হবে। হ্যাভ টু কিল দিস অফেন্ডিং এজেন্টস, দ্যাট মে বি ব্যাকটেরিয়া, দ্যাট মে বি ফাঙ্গাল, রিমেম্বার দ্যাট মে বি একটা ফাঙ্গাল এঁকেই দিলাম, একটা ফাঙ্গাল, ব্যাকটেরিয়া অ্যান্ড অলসো ফাঙ্গাল। Oxygen is there. Now this oxygen is formed as a কি বলে এটাকে, superoxide, হ্যাঁ theoretical, this is called superoxide. In short আমি বলে দিই, সব এগুলো বই আছে। Then it will form H2O2 by a H plus, and then it will form HOCl. This is a last target products that is called hypochlorous acid, that means bleach, তাই তো, এটাকে বলা হয় ব্লিচ, BLEACH, ব্লিচ। নাও এইখানে একটা এনজাইম, নাম্বার 2 তে একটা এনজাইম, নাম্বার 3 তে একটা এনজাইম ইজ দেয়ার। ফর্মুলা ইজ NSM, NSM ফর্মুলা। তাহলে ফার্স্ট হচ্ছে N, that means this is NADPH oxidase, NADPH oxidase. Number 2 S, বল, superoxide dismutase, superoxide dismutase. Now M is myeloperoxidase, myeloperoxidase. এখানে অনেক কিছু হচ্ছে, অনেক এভরিথিং। যদি এইখানের মধ্যেই ঢুকে যাই তাহলে ক্লাস ফিনিশ যাই করা। তাও গ্রসলি আমি কিছু কিছু জিনিস দিচ্ছি বলে, হোয়াট আর দ্য ইম্পর্টেন্স ফর ইয়োর এক্সাম অ্যান্ড অল অ্যাজ ওয়েল এজ নলেজ অ্যান্ড ক্লিনিক্যালি অলসো ইম্পর্টেন্ট। সো অক্সিজেন, NADPH oxidase taking the one electron, superoxide formation, highly toxic, and then by the help of superoxide dismutase, H2O2 peroxide, hydrogen peroxide is formed, less toxic. Again by the enzyme myeloperoxidase to the HOCl, that is bleach, and these ultimately damage করে একদম এটাকে disintegrations দেবে। Disintegrations are damage all the cell walls of the bacteria and fungal and damage and kill the bacteria and fungus. HOCl finish. In fact these also can damage, right? এটুকু বোঝা গেছে? So oxidation, extra electron, taking extra electron, superoxide by the enzyme NADPH oxidase, then the superoxide dismutase, then the myeloperoxidase, NSM. Now suppose a patient, যেমন আমার কাছে এসেছিল একটা জলজ্যান্ত উদাহরণ হচ্ছে আমার কাছে আছে, a child having age 8 months basically, 8 months with history of recurrent chest infection, taking antibiotics, and really pneumonia is happening, seeing the X-ray, recurrent pneumonia, and also formation of different types of abscesses all over the body, different abscesses, ফোঁড়া হচ্ছে, abscess all over the body. When the child presented to me after around 3 to 4 months of giving the X-ray, antibiotics, different tests of there, then come with the abscess, only came with me abscess. I thought this is the abscess, no problem, the antibiotic given and will cure. Okay, it is cured, then again come after 1 week with the chest infection. and these are recurring and recurring is that. So my thought was that some immunity depression is there. I go for a HIV testing because his father is a truck driver. So I thought some relation must be there in the family, whether he is carrying HIV or not. So I done that. That's also came out a negative. But leukocyte count is persistently high. That is more than 11,000 always, giving after the antibiotics also. That means that is a clue that neutrophil have to understand what is happening here. That means neutrophil is unable to kill the bacteria by this process because it's happening in the abscess, recurrent pneumonia, recurrent chest infection. There must be some infection, maybe viral, maybe bacterial. But if it is viral, then the leukocyte count will be less. There must be leukocyte count is less. But neutrophil count is high above, total count is showing the high, lymphocyte count is little bit in higher, in the lower range. So what is happening? Neutrophil is a culprit. Now I thought the different stages of some history is taking. আগে আমি কিছু history নিইনি, past history বলেছি ওরা তেমন বলেও নি। তারপরেতে I have seen 3 months ধরে ওর এইরকম গোছা antibiotics prescriptions are there and the baby is taking antibiotics almost every week. That is the important clue for the chronic granulomatous disease. That is the important clue. That is the chronic granulomatous disease, CGD, chronic granulomas formations is happening in the baby boy, basically boy was there, is a 8 months of age, recurrent chest infection. How it is happening? Here, main culprit is here, deficient of that child, that is inherited, congenitally deficiency is there. Now he or she is exposing more and more in outside, so now getting bacterial infection is there. We are here always, we came across with bacteria virus always. এখনো তোরা কেউ এক্সপোজ করে এসেছিস, খেয়ে এসেছিস বা কিছু করেছিস, হাত দুসনি, মুখে হাত দিচ্ছে, দিচ্ছে মুখে হাত দিচ্ছে, so this taking is there. So in this way we are gaining microbes, but our body is so much strong, the immune system is so much strong, it is the fast acting for the immunity system, and so this activates the immune system to deactivate or kills that microbe. In this baby, how they can kill? Because the first enzyme NADP oxidase is absent. Now how superoxide will form? How hydrogen peroxide will form? How HOCl will form? That means the baby is unable to kill the bacteria, but their neutrophil function is right. Neutrophil to ঠিকঠাক আছে, enzyme টা নেই, because there is some mutation. That is mostly enzyme deficiency mean autosomal recessive. এটা খেয়াল রাখ, mainly 99.9 cases, in case of any deficiency of enzyme, that mostly it is autosomal recessive. That does not mean that X-linked হবে না, হতে পারে এটা X-linked. If it is X-linked, then only male presentation will be there. X-linked dominant, especially X-linked dominant. So this NADPH oxidase is deficient, autosomal recessive patient, and then came to the history. যে হ্যাঁ ও দাদুরও এরকম বারবার ইনফেকশন হয়ে তাকে অ্যাবসেস হয়ে ফোড়া হয়ে আলটিমেটলি সেপসিস হয়ে মারা গেছে। So that is important history. I don't know the history, but chronic granulomatous disease is caused by the deficiency of this NADPH oxidase. That is the clinical implications of the oxidative burst or the respiratory burst. So this is happening here. If it is not formed, if it is not there, it will not form. It will not form, then it will not form. No formation of this and no damage or kill this. তাহলে করছেটা কি? করছে কিচ্ছু না, এই যে ব্যাকটেরিয়া বা ফাঙ্গাল আছে, এই চারিপাশে নিউট্রোফিল এসে ওকে ঘিরে দিচ্ছে যাতে দে ক্যানট স্প্রেড টু আদার বডি। বডি ইজ ট্রাইং to accumulate the living bacteria inside a place, and these ultimately forms a granuloma. Sometimes is from the abscess, and that's why the chest infection is there, forming the different types of granulomas in the lung, and the patient is suffering from fever, etc., etc. Treatment খুব সাংঘাতিক. I advise them the basic treatment is that stem cell transplantation. The patient is unaffordable, say তারা afford করতে পারবে না, so I am giving them a long term antibiotics. I am giving them long term antibiotic. Long term antibiotic prophylactically cotrimoxazole for the antifungal, and also sulfathrin, trimethoprim is there, it is given. But কদিন দেব? That will come into the resistance. So you have to check some bone marrow transplantation from NGO. আমি তাদের কিছু অ্যাড্রেস দিয়েছি, যদি করে কেন? If it is the bone marrow transplantation, that means remember myeloid series, hematopoietic stem cell, that is from where neutrophil is coming, and neutrophil having the NADPH oxidase, new NADPH oxidase of new neutrophil may appear. তাহলে different types lineage-টাই চেঞ্জ করে দেবো. Those neutrophil is coming from this series and lineage, from the fresh stem cell transplantation, fresh neutrophil having the NADPH oxidase enzyme, and they will perform well like that. If NADPH oxidase is present, then there is no problem at all. Now, there are some bacteria, they produces the catalase. তোরা সবাই কেউ না কেউ কোনো এসছিস বা তোর ফ্যামিলি এসছিস, there is dreaded bacteria is a Staphylococcus aureus. Staphylococcus aureus is very common, it's not uncommon. We পাই regularly পাই. These bacteria and others, many bacteria are there, and any fungus are there, that is the catalase positive. That means this bacteria releases the catalase. These bacteria are producing the catalase. এই একটা ব্যাকটেরিয়া is there. এই দেখো কি বলছে, দেখতে পাবে না তার মধ্যে ওরা, তাই তো অসুবিধা হচ্ছে। হ্যাঁ, বন্ধ করো এইটা। হ্যাঁ, so this bacteria is producing catalase enzyme আস্তে। so this catalase enzyme এর কাজ কি? catalase কিচ্ছু করবে না, ও এসে চুপচাপ এসে এই কার্বন হাইড্রোজেন পারঅক্সাইডে অ্যাকশন নেবে, then it will form benign water and O2. তাহলে ক্যাটালেস কি করবে না, আমার H2O2 কে ভেঙে H2O plus O2 করে বেরিয়ে যাবে, benign. So how we can damage by to kill the bacteria by formation of the HOCl, that is the bleach, that is the more strong, কারণ এ তো চলে যাচ্ছে এইদিকে, ক্যাটালেস ওকে ভেঙে দিচ্ছে, so very difficult to manage by our immune system. সেই জন্য আমাকে দিতে হয় অ্যান্টিবায়োটিক 5 দিন পরে, বলেছিলাম। Sometimes it happens that Staph aureus infection suspected. If I suspect in the first or second day, I always recommend antibiotic in the first day. Only the exceptional case, that is when if I suspect that yes, that may cause due to the Staphylococcus aureus, because but this bacteria, it produces the catalase, and the body immune system, like the oxidative burst, may be hampered by the deactivating the hydrogen peroxide, and further step, that is the hydrogen peroxide to HOCl will not be occur. Then bleaching procedure হবে না, কতটা করতে পারে, যেটুকু সুপারঅক্সাইড তৈরি হলো, if NADPH oxidase is present. But suppose remember, you can think that when chronic granulomatous disease patient is suffering from Staph aureus, dangerous, that may cause sepsis, septicemia, multi-organ failure and death. এই পেশেন্টে যেটা কথা বললাম, if he is not treated properly or ultimately stem cell transplantation, he will die within basically years. Because recurrent chest infection, recurrent Staph aureus infection will be there, and resistance will come on with the medicines chronically I am prescribed. যতদিন আছো বেঁচে এটা খাবে, ইমিউন থেরাপি ইজ দেয়ার, এনজাইমে কোনো আলাদা করে কিছু পাওয়া যায় না যে আমি এনজাইমটা দিয়ে দেবো, নো, বিকজ দিস এনজাইম ইজ প্রেজেন্ট in the this way is coming into the neutrophil. So in the phagosome formation যেটা আমরা এই যেটা করেছি আমি, সেটা হচ্ছে আমার মেমব্রেন অফ দ্য নিউট্রোফিল, তাই না? এটা হচ্ছে আমার মেমব্রেন অফ নিউট্রোফিল। এইখানে আমার যখন ব্যাকটেরিয়া ঢুকছে, দেন when the load of bacteria is much more high, then the phagolysosome করছে, but phagolysosome is unable to kill the most of the bacterial loads, and then oxidative function will come into action, and this action come by the formation of the HOCl, that is a bleach, it is called bleach, that kills the bacteria. But if catalyst producing bacteria, catalyst producing fungus, they convert the hydrogen peroxide into the very benign water and oxygen, and so the further step HOCl bleach will not form. That's why this is dangerous. Now, interesting thing is that suppose this one is not coming due to superoctane dismutase নেই, হতে পারে যেমন NADPH অক্সিডেজ নেই, তেমনি সুপার অক্সাইড ডিসমিউটেজটাই নেই, বা NADP অক্সিডেজটা নেই, তাহলে হাইড্রোজেন পারঅক্সাইডটা তৈরি হবে? নো। Now interesting thing is that like our cell, our own self cell, bacterial cell also produces hydrogen peroxide. Bacterial cell also produces hydrogen peroxide. Interesting. The bacteria hydrogen peroxide তৈরি করবে, and our body utilizes their hydrogen peroxide to this conversion. বুঝতে পারছিস? ব্যাকটেরিয়া সেল H2O2 তৈরি করতে পারে আমাদের মতো, আমরা যেমন এনজাইম দিয়ে করলাম, হ্যাঁ, সুপারঅক্সাইড ডিসমিউটেজ দিয়ে, সুপারঅক্সাইড থেকে সুপারঅক্সাইড ডিসমিউটেজ দেন H2O2 after formation of H2O2 hydrogen peroxide, then only it will happen to formation of the HOCl, that is bleach and important for the kill of the bacteria and fungus. সুপারঅক্সাইড ডিসমিউটেজ নেই, is absent, আমার মনে কর নেই, আমি কিন্তু ওকে টুকটু করে মারবো, কেন, কি করে, how, if bacteria is enters, every bacteria or fungus may produce H2O2 to some extent হলেও, I utilize, my cell will utilize this H2O2 in this level, and then it converts into the HOCl because myeloperoxidase enzyme is present. Interesting? বুঝতে পারছিস? So in this way our daily immune system is happening. Yes, any problem? বেরোবার জায়গা আছে রে ভাই, যখন ক্যাটালিস্ট বের করতে পারছে, নিশ্চয়ই H2O2 বের করতে দেবে তো, সিক্রেট করবে, হ্যাঁ, সবসময় করছে। আমার যখন আছে তখন ইউটিলাইজ করছি না, আমার যখন নেই, I have some sorts of superoxide dismutase enzyme, আমার মিউটেশন আছে, আমার নেই, we are not getting H2O2, then we will try to form, taking their H2O2 to form the HOCl, because H2O2 hydrogen peroxide is essential for the formation of the HOCl. HOCl means bleach. মাথা মধ্যে ঢুকিয়ে নে, ব্লিচ মানে কি, আমার সেল মেমব্রেনকে পুরো একদম শেষ করে দেবে, ড্যামেজ all the cell membranes, right? So in this way this oxidative burst functions. Now the one questions may you ask, that if it is the neutrophil, that so much toxic material is forming in the oxidative burst that can damage our own cell also. Yes, করতে পারে, ডেফিনেটলি পারে। তার জন্য কিছু কিছু ব্যবস্থা আছে। কি কি আছে? তার জন্য ব্যবস্থা হচ্ছে আমাদের H2O2টা হচ্ছে আমাদের একটু less toxic and also unstable, and so if it is bacteria present, then it will not form further HOCl. Neutrophil also not activated to produce the myeloperoxidase enzyme, and so HOCl does not form, and H2O2 formation is there. This is a less toxic. Now we come to the point. Come to the point means, suppose this is H2O2, it can go in two ways. Again, one is with reaction with the iron. সেই তোকে বলছিলাম না যে আয়রন ফ্রিলি থাকতে পারে না, ফ্রিলি আয়রন থাকতে পারে না। দিস ইজ কল্ড ফেন্টনস রিয়াকশন, আসবে তোদের বইয়ে। অ্যান্ড অলসো is formed in another way, that is hydroxyl. Hydroxyl is also a very toxic, very toxic. When you are exposing in the radiation, মনে করো রেডিয়েশন একটা হচ্ছে ওখানে typically, then it will form into hydroxyl, UV light, hydroxyl ion, and that can damage your cell. So if it is free iron is there, so that is always in our body, always make a maneuvers, make a system that at least we can't allow free iron inside our blood, because Fenton's reaction করে OH মানে hydroxyl ion তৈরি করে দেবে, hydroxyl ion, hydroxyl, yes, and that will damage our cell. So that must be prevented by what process? By the glutathione reduced form, glutathione reductase. এগুলো বায়োকেমিস্ট্রিতে আছে, আমি জাস্ট একটু ঢুকে আবার বেরিয়ে আসবো, জাস্ট এইটুকু মনে রাখ। When hydrogen peroxide is forming during the damage of the bacteria, then the glutathione's reduced form is helping us to forming the H2O2 and water that is safe. And this reduced form of glutathione is also essential for the RBC glycolysis. পড়া হলে পারবি বুঝতে, আমি ওইদিকে যাচ্ছি না, is the correlation is there, yes. So NADPH is essential for the again reduced form of glutathione. If glutathione reduced form is utilizing and it is exhausting, then the further step will not occur. So glutathione reduced form must be there by some maneuvers, by some mechanism, that is NADPH. That again is come to the reduced form, again it is hydrogen peroxide to form a water into oxygen, and that is benign. And that's why our system, our body helps to protect ourselves, our body. বোঝা গেছে? So অল্প ব্যাকটেরিয়া, ফ্যাগোলাইসোজোম, একটু বেশি হয়ে গেল অক্সিডেটিভ বাস্ট, NSM, oxygen superoxide formation, then hydrogen peroxide, then bleach. Then bleach will kill all bacteria and fungus. If it is bacteria is producing catalase, that means catalase producing bacteria like Staph aureus, then it will break the hydrogen peroxide into water, into oxygen. That is benign. So those bacteria are dangerous for our, because if hydrogen peroxide breaks out, then further HOCl formation will not occur. If it is not occur, then the bleaching procedures to kill the bacteria and fungus will not occur. If the bacteria is no our enzyme is absent, also suppose NADPH oxidase আমার absent আছে, তাও সে হাইড্রোজেন পেরক্সাইড কে ইউজ করে HOCl তৈরি করতে পারবে খানিকটা হলেও। কিভাবে পারবে? To from the bacteria is taking their H2O2, কেমন নিজের বাসটা নিজেই নিচ্ছে, H2O2 বের করে দিল নিজে, ও জানে না বডির এরকম মাইলোপেরক্সাইড এনজাইম বসে আছে ওর জন্য। So myeloperoxide come connected in the H2O2 and then formation HOCl by binding with the chlorine. So in this way the bleaching procedures it come where the enzyme is absent or present. Body, my fundamental take home message is that our body always try to destroy the microbes which is entering day by day. Whenever we take antibiotic, that means so much immunity system is highly active and the mechanism is now to some extent failure and load of bacteria is too much high that may cause septicemia further. Amra bhoye in fear we can use antibiotics. Otherwise our immune system is so much intelligent and so much active. Two types of immunity are there. There is the innate immunity, one is the adaptive immunity. পাচ্ছি শুনতে আওয়াজ আসছে তো? ঠিক। So if it is innate immunity, that means fast acting. এই যে তোরা এখন হোস্টেলে you have taking the breakfast and কেউ হাত দিয়ে খাসনি, কেউ মানে হাতে আংটি পরে আছিস, কোথায় হাত দিয়েছিস তার কোনো ঠিক ঠিকানা নেই, and the innate immunity is on. innate immunity. That means innate immunity compromise the cell, organ, system, chemicals like neutrophil, macrophage, monocyte, dendritic cell, NK cell, natural killer cell, mast cell. These are the soldiers of the innate immunity. But adaptive immunity is a specific and for the importance the immunologic memory for that immunity. I will come later. So now the newer topic. Time আছে দেখি. So newer topic আমাকে সব বললে মারতে আসবে সব স্যাররা, কি সব বলছে, নেটোসিস বলছে. So now if a bacterial load is too much high, এত বেশি হয়ে গেছে যে থাকা যাচ্ছে না, আইসিইউতে আছে, কোনো একটা ইনফেকশন হয়েছে, সাপোজ নট অনলি ব্যাকটেরিয়া, is recently proved that is a COVID also. So much load is infecting agent is too much high, and neutrophil does not entertain in a viral infection as such, but in COVID infection it happened already. What is happening? Neutrophil has lobes, right? Lobes are there. Lobes means nuclear lobe. In the nuclear, suppose এটাকে আমি একটু বড় করে আঁকি, there is a chromatins, DNA structure আছে, chromatins are there, right? And then chromatins means DNA structure means arginine, lysine. পড়ছিস তোরা একটু করে. If arginine is present, then a specific reaction happens during the dying process of neutrophil. যখন বয়স হয়ে গেল নিউট্রোফিলের বা ব্যাকটেরিয়াকে মেরে সি ইজ এক্সস্টেড. নাও ওয়ান ইম্পর্টেন্ট টার্ম ইজ সিট্রুলাইজেশনস, মনে রাখার দরকার নেই. আর্জিনিনটা ইজ ট্রান্সফর্ম ইনটু দ্য সিট্রুলিন. এইখানে যে আর্জিনিন থাকে, আর্জিনিন ইজ ট্রান্সফর্ম ইনটু সিট্রুলিন, and this citrulline, C for C, this decondensation of the chromatin, decondensation of chromatin. Chromatin তো এখানে ছিল, এই যে chromatin নিউটক্স থাকবে, নিউক্লিয়াস থাকতেই হবে. So decondensation means কি, ভেঙে দিচ্ছে, ব্রেক করে দিচ্ছে, এগুলো ব্রেক এইগুলো আমার যা আছে সব ব্রেক করে দিচ্ছে, সব ব্রেক সঙ্গে এখানে গ্র্যানুলস ইজ দেয়ার. So decondensation is happening by a process that is called deamination. আমি ডিটেলসে মুখস্থ রাখতে বারণ করছি, টার্মগুলো বলছি কারণ ইনস্ট্যান্টলি আসছে তাই. So this arginine in a nuclear part, it is transformed into the citrulline, and then decondensation of the chromatin inside the nucleus is happening during the bacterial load. এখন মাথাতে রাখ, bacterial load is high, hugely high. Target কি থাকবে? To restrict the spread and to kill those load, bacterial load, মারতে হবে. Then what is happening? This condensation is happening inside the neutrophil, and then this decondensation is chromatin, and then these chromatins will come. These chromatins will start like a meshwork of chromatin. একটা জালি তৈরি করবে chromatin. That is proved. কোনোটাই মানে ভার্চুয়াল নয়. This will come out. That is called neutrophil extracellular traps. এটা একটা trap for what? Trap for কি এগুলো? Yes, these are the bacterias. Nodes আছে, ধরলো বেটাকে. So these are the bacteria come into those areas after the decondensation of the chromatin or the arginine to citrulline. It is called citrulization, and also the deamination, and after that this is called neutrophil extracellular cell বাইরে traps. কেন? To trap those bacteria to decrease the spreading, as well as these granules and granular product will come here. এখানে myeloperoxidase, cathepsin সব কিছু আসবে, ইলাস্টেজ সমস্ত আসবে, and they will damage. পেয়ে গেছে ব্যাকটেরিয়াকে, পেয়ে গেছে ওখানে, they will damage and trying to reduce the load of bacteria. There's a very, very new concept, and this is a going on research. And so these are the neutrophil extracellular traps is one of the mechanism to fight our bacteria. Now question is that it is always beneficial? No, কেন? চিন্তা কর, ইনসাইডে আমার সেলুলারের ডিএনএ আছে, ডিএনএ এর ক্রোমাটিন অলওয়েজ মাস্ট বি দেয়ার ইনসাইড দ্য সেল। সে কি করলো? ডিকন্ডেনসেশন করলো, করে সমস্ত নিউক্লিয়ার মেটেরিয়াল, দিস আর দ্য নিউক্লিয়ার মেটেরিয়াল কামস আউট ফ্রম দ্য সেল। আওয়ার বডি মানে আমাদের সেল recognizes that nuclear antigen as a foreign. That's why may cause autoimmune diseases like systemic lupus erythematosus, SLE, মেয়েদের হয়, SLE, systemic lupus erythematosus. Now it is on research, if this netosis, this formation, if reduced, then we can, yes, systemic lupus erythematosus, SUS, আমার লেখাটা খারাপ, sorry for that, systemic lupus erythematosus, SLE, বা simply মনে রাখ lupus, it is also called lupus. If you tell the lupus, that that's directly significant that you are telling that systemic lupus erythematosus. So this is an important clue point. That is a neutrophil extracellular trap does not always helpful. It is helpful for the bacterial load to trap the bacteria and kill the bacteria and reduce the spreading. But in the same time, as the chromatin is a nuclear content is coming outside of the cell, that means our body, কখনো দেখেনি তো, our body cells is not recognized that before, এটা পাইনি, that means he detects as a foreign. তার নিজেরই সেলকে নিজেই যদি অ্যান্টিজেন বলে, that is called autoimmune disorder, and this forms the auto antibodies against that, and that will create a problem, and that is the term is called anti-nuclear antibody. সেদিনকে একটা বাচ্চা মেয়ের আমি করলাম due to this rash. The lupus is presentation is a specific, in the cheek there will be a rash, specific rash, that is ANA, anti-nuclear antibody is too much high of that result. That means a suspected case of lupus. That may be due to this recurrent infection. There is history of recurrent infection is there. Extracellular traps are there. Nuclear content come out of the cell, and then this invites the autoimmune antibody formation, and this lupus is happening in the clinically. Also in gout also. In the gout, monosodium urate is a inviting agents that inflammation বাড়ায়, inflammations invite করবে, and it also augments the formation of the neutrophil extracellular traps. And that's why we always use, in case of gout, that is the colchicine, anti-inflammatory drug. লিখবার দরকার নেই, colchicine একটা anti-inflammatory drug. So I want to treat the gout first as a anti-inflammatory drug to suppress these neutrophil extracellular traps. That means take up message is that neutrophil extracellular traps is a new concept of trapping the bacteria by the decondensation of the chromatin and forming the meshwork of the chromatin, and also granules are there to deactivate or destroy the bacteria is good. But if it is much more formation is there, then our body detects that nuclear content as a foreign, and that's causing the invitation of the autoimmune body formation, and that's causing the lupus. That is means a autoimmune disorder will be there, and so on. And also in gout also, we always try to reduce the formation of the nuclear. Research is going on. যদি কখনো থাকে, যদি যা বেরোবে আমি বলে দেবো। So in this way we can finish this. This is a new topic, but it is interesting for you because you are going to be a doctor. Your knowledge should be sound and up to date, and till date আজকে পর্যন্ত যা যা knowledge থাকবে নিউট্রোফিলের, you have to know. কতটা মনে রাখতে পারবি, that is not a problem, বাট অন্তত এইটু থাকবে যে হ্যাঁ নিউট্রোফিল এক্সট্রাসেলুলার ট্র্যাপস বলে কিছু হয়, মুখার্জি কিছু একদিন বলেছিলেন। নাও এত তো গল্প হচ্ছে মাইক্রোবস। নাও প্রবলেম ইজ দ্যাট সেলফ অ্যান্ড নন সেলফ, এই যে একটু বলছিলাম সেলফ অ্যান্ড নন সেলফ বুঝবো কি করে? আওয়ার বডি কি করে বুঝছে যেটা হ্যাঁ এটা আমার সেল আর এটা অন্যের সেল? সাপোজ এ ব্যাকটেরিয়া এন্টার্স, সাপোজ এ ব্যাকটেরিয়া না একটা মাইক্রোবস, দিস ইজ এ মাইক্রোবস। Remember always, whenever you have to detect or phagosome or anything attraction of the bacteria in front of neutrophil, macrophages, monocytes, NK cells, there must be some recognition receptor on those cells, as well as some molecular patterns are there in the microbes also. Suppose this is a microbes, these are the molecular patterns are there, এটা ফ্ল্যাগ করে গেছি কিন্তু molecular patterns are there and this is repeated molecular pattern are there, and that is called, these are called pathogen associated molecular pattern, PAMPS, pathogen associated molecular pattern। পাচ্ছিস তো লিখতে খুব সহজ, প্যাথোজেন অ্যাসোসিয়েটেড, এটা একটা মাইক্রোবস, দ্যাট মিনস ইটস এ প্যাথোজেন, তাই তো? মাইক্রোবস মিনস ভাইরাস মিনস ইটস এ প্যাথোজেন। সো প্যাথোজেন অ্যাসোসিয়েটেড মলিকুলার লেভেলে এটা আছে, এটা ইলেকট্রন মাইক্রোস্কোপে গিয়ে এইরকম একটা পাওয়া গেছে মলিকুলার প্যাটার্ন, দিজ আর প্রেজেন্ট ইন দ্য কি প্যাথোজেন্স, রাইট? নাও in our cell innate immunity soldiers কে আছে? নিউট্রোফিল, বেসোফিল, ইওসিনোফিল সবাই আছে। তো ইওসিনোফিল, বেসোফিল আর নট দ্য ফ্যাগোসাইটিক, বাট দে আর ইন কেস অফ অ্যালার্জিক, কোনো একটা পোলেন্স ঢুকেছে, দে উইল ওয়ার্ক। হেলমিনস অত ফরেন্ট, দ্যাট উইল অ্যাক্ট। আমি আগের দিন ক্লাসে এগুলো বলেছিলাম, ইওসিনোফিল হাউ। সো প্যাথোজেন অ্যাসোসিয়েটেড মলিকুলার প্যাটার্নস আর দেয়ার, অ্যান্ড pattern recognition receptor. Suppose this is a neutrophil, and these are the key PRR. What does mean? Pattern. এই যে প্যাটার্নটা, এই পিটা এইখানে নিয়ে এলাম। প্যাটার্ন রেকগনিশন মানে কি? রেকগনাইজ করা, চেনা। ইয়েস, তো নামগুলো দেখলেই হয়ে যাবে। এইটা হচ্ছে প্যাটার্ন রেকগনিশন রিসেপ্টর, হচ্ছে মাথায়। তাহলে এমন নয় যে ঢুকলেই ও দুম করে গিয়ে খেয়ে নিল, তা নয়। আগে তো চিনতে হবে ভাই তুই কে, তা চেনার জন্য প্যাথোজেনের মধ্যে থাকতে হবে প্যাথোজেন অ্যাসোসিয়েটেড মলিকুলার প্যাটার্ন। These are the pattern, these are formed in the, like a protein molecules are there, and that is genetically, that is imprinting us there and genetically controlled, and also the receptor, pattern recognition receptor on the our innate cells বা innate soldiers, innate immunity soldier. Immunity means আজকে এইটুকুই থাকবে মনে, innate immunity and adaptive immunity. Innate immunity is a non-specific, যাকে পাচ্ছে তাকেই খাবে, ইনস্ট্যান্ট খাবে, and they will feverishly fast. এই কথাটা ইম্পর্টেন্ট, feverishly fast, ফিভার আসছে, তাই তো? কারো আসছে তো ফিভার এখন? এই যে ইননেট ইমিউনিটি অ্যাকশন নেয় বলেই ফিভার আসে। তাই ফিভারকে সাপ্রেস করার জন্য প্যারাসিটামল, আনটিল আনলেস ইউ আর আনকমফোর্টেবল, ডোন্ট গিভ অ্যান্ড ডোন্ট ইউজ ইয়োরসেলফ অলসো। Fever is coming for that means our innate immunity is functioning. They detects the microbes or agents or pathogens enter in our body, and they are trying to kill those. And that's why this feverishly fast come from the fever. That's why fever term has come. The feverishly come, presentation will be fever, and that is the innate immunity. There's another chapter. Now this is the pathogen associated molecular pattern, and pattern recognition receptor are there. Now, what are the types of প্যাটার্ন রেকগনিশন রিসেপ্টার। অত তো সহজ নয়, কেউ আসবে ব্যাকটেরিয়া, কেউ আসবে ফাঙ্গাস, ভাই আমি বুঝবো কি করে? হ্যাঁ, you have to understand. So PRR mainly are four types. কি কি? আগে তোরা শুনেছিস টোল লাইক রিসেপ্টার্স, শুনেছিস নাম? শুনিস নি? টোল লাইক রিসেপ্টার তো আমরা এইচএসএ পড়তাম রে। ওই ড্রসোফিলিয়া একটা ফ্লাই ছিল, ফ্রুট ফ্লাই, হ্যাঁ, দ্যাট ইজ এ টোল প্রোটিন ইজ দেয়ার, ফ্রম হোয়ার টোল রিসেপ্টার ইজ কাম, হ্যাঁ, ফ্রুট ফ্লাই ছিল না একটা, কি নাম ছিল একটা, ড্রসোফিলিয়া মেলাসিজিয়ান, হ্যাঁ হ্যাঁ ওই যে কি সব বলছে। So these are the toll-like receptors, number one. Number two is a C-type lectin receptor. C-type lectin receptor. Number three, আগে নামগুলো বলে দিচ্ছি, rig receptor. rig, R-I-G, বেসিক্যালি rig লাইক বললে ভালো হয়, আমি বললাম কেটে দিচ্ছি এটা। Rig right receptor, rig for RIG. I am coming in details. 4 is a NOD, like receptor. NOD, N for NOD. নামগুলো খুব কঠিন নয়, বাট অন্তত জেনে রাখ এইটা যে there are 4 types of pattern recognition receptors are there. Different actions for there. Suppose toll like receptor. In human, there are 10 types of toll like receptors until now. ঠিক আছে, নাও টোল লাইক রিসেপ্টর 4 আমি বোঝাবার জন্য বলছি, মনে না রাখলেও হবে। ইট রেকগনাইজেস অনলি দ্য লাইপোপলিস্যাকারাইড অফ দ্য গ্রাম নেগেটিভ ব্যাকটেরিয়া। বুঝতে পারছিস? দিস রেকগনাইজেস দ্য গ্রাম নেগেটিভ ব্যাকটেরিয়া। হাউ? রেকগনাইজেস দ্য লাইপোপলিস্যাকারাইড গ্রাম negative, is not positive, is gram negative, because lipopolysaccharide is present only in the surface membrane of the gram negative, remember. That's like toll-like receptor 378 only detects virus, TLR 378 only virus. তেমনি TLR 5 only detects the flagellin, কোথায় থাকে? ব্যাকটেরিয়াল ফ্ল্যাজেলিন কে ডিটেক্ট করে। বুঝতে পারছিস? দ্যাট মিনস প্যাটার্ন রেকগনিশন রিসেপ্টর ইজ রেকগনাইজিং দ্য পার্টস অফ দ্য ব্যাকটেরিয়া, ভাইরাস এটসেট্রা। অ্যান্ড অলসো টোল লাইক রিসেপ্টর 2 এ কি করবে? গ্রাম পজিটিভের পেপটিডোগ্লাইকেন, লাইপোগ্লাইকেন, দ্যাট উইল বি ডিটেক্টেড। আমি নামগুলো বললাম না। সো 10 টাইপস অফ টোল রিসেপ্টর দেয়ার। It is a pattern receptor. Toll-like receptor short note দেয়। Toll-like receptor short note দেয়। This is a pattern receptor recognition. A pattern recognition. ওই দেখ আমার হয়ে গেছে। That means 5 minutes more. So this is the 4 types are there. That is the C lectin receptor, rig like receptor. Time permit করলে বলছি, not like receptor. Okay. So C type lectin receptor only detects the কথা বলবি না, carbohydrates of the bacteria, carbohydrates parts only. Rig like receptor, this is a soluble pattern recognition receptor. Soluble means cytoplasm-এর ভেতরে থাকবে, soluble, these are soluble. Rig means retinoic acid inducible gene 1, মনে রাখার ইয়ে নাই, retinoic acid inducible gene 1. রিগ, RIG, নডেরও ফুল ফর্ম আছে, নিউক্লিওটাইড অলিগোমারেশনস ডোমেন, দরকার নেই। জাস্ট রিমেম্বার প্যাটার্ন রেকগনিশন রিসেপ্টার্স আর 4 টাইপস and they recognizes the specific parts of the bacteria and viruses. Enough for you. যদি কোনো হায়ারে যেতে চাও তখন এগুলো তোমরা যাবে। So until now this is very much interesting that you already detects, understand the oxidative burst, netosis means ওই প্রসেসটাকে বলা হয়, that is process that form the neutrophil extracellular traps, the process formation by the decondensation of the chromatin, it is called netosis, just like apoptosis. ঠিক আছে? So in this way we can day by day improving for to destroy our microbes, but unfortunately last 20 years no antibiotics has been discovered. So until unless specific requirement of antibiotic is there, don't prescribe unnecessary, irrationally antibiotics to your near and dear ones as well as your patient. Until unless you accept the patients like your near and dear ones, you will be not a good practitioner at all. So whenever you are prescribing any antibiotic in future life, always search whether it is a dite hobe ki dite hobe na. It is already discussed that our body system, immune system is too much active. If need, we have to do some chronic granulomatous disease. I explained about my patient also, the how it is due to the NADPH oxidase deficiency is there. So that's why it's happening. It is a inherited and genetically mutation is there. So this patient may come with a recurrent chest infection, recurrent granule formation, recurrent abscess formation is there. So always take a history, and neutrophil count is very important for that. And also the neutrophil extracellular trap. This is a very new concept, very new concept for last 1 and basically 2 years. Basically after the COVID also it is discussed that the one important thing, last line, that is a COVID infection. That is we already told that in the March April 2021 the second wave, there's a too much thrombosis is happening. We lost many doctors due to the refractory hypoxia. Oxygen dio kaj hocche na. That's happening due to this formation of thrombus, pulmonary thrombus formation was there, and the thrombus formation is due to neutrophil extracellular traps and trapping the platelets also. Trapping the platelets also in that time, and that is why D-dimer level, CRP level is going high and high. It means NLR, ওই সময় আমরা দেখছিলাম neutrophil and lymphocyte ratio, and NLR ratio, those patients who lost their doctors and relatives also, NLR is too much high. That means neutrophil level খুব high. COVID is a virus. How neutrophil can rise? Questions? Due to this formation of this netosis is happening. So that's why anti-inflammatory drug, that is a steroid, was given, and to some extent this is a rewarding, but not all the cases. So refractory hypoxia in case of COVID infection also is a role of this new concept, that is a neutrophil extracellular trap. Thank you very much. Dismiss my class. Thank you.
What is your practical today? Constituents of abnormal constituents of. What are the abnormal constituents present in the urine? Out of that, what test we will do today? Only two tests. One is test for reducing sugar, test for reducing sugar. What is the test name? Benedict test. Good, Benedict test. Another test, heat coagulation test. Test for protein, that is heat coagulation test. Also Hello test is there, and another test is that is sulfosalicylic acid test. Okay. So first, what is the Benedict test procedure? Benedict test procedure. 5 ml of, you will take 5 ml of Benedict reagent in a test tube. Then you will add
8 to 9
8 drops, 8 drops or 10 drops. Okay. 8 or 10. 8 drops of urine. Then you properly mix it. After mixing, you will heat. How much time? 1 to 2 minutes. 1 to 2 minutes you will heat. Then what you will get? You will get.
Sir, a range of color.
Okay. It may be green, GYOR. Okay. Vibgure is there. VIB got it. Okay. GYOR. It may be green. If it green color, then it is also positive. If it is yellow color, that is also positive. If orange color, that is also positive. If brick red color, that is also positive. Okay. So any color will come, that is positive. So if suppose green color came, then what is the gram percentage?
0.5.
0.5 to 1 gram percentage of sugar present in the urine. Suppose yellow color 1.1 to 1.5, and if it is orange color 1.5 to 2. If it is brick red, that is more than 2 gram per dl. Okay. And what is the gradation? Suppose green color, what will be grade?
Negligible.
What is average? Grade, that is plus 1, 1 plus gradation. Okay. If yellow, then it will be 2 plus If it is 3 plus, then it will be orange. If brick red, that will be 4 plus. That is your gradation, and all these things are there. What is the gram percentage? That is your amount of sugar present in the urine. Amount of sugar or concentration of sugar present in the urine. Okay. From that one question can be derived, that is why Benedict test is called as semi-quantitative test. Why it is called as semi-quantitative test? Why? You can tell semi-quantitative test. It is a qualitative test. You are seeing the color, so it is a qualitative test. But you are telling yes, Benedict test is called as semi-quantitative test. Why it is called semi-quantitative? Not fully quantitative. It is semi-quantitative. Why it is called semi-quantitative? Because by seeing the color of the solution, you can assume how much gram percentage of sugar present in the urine. You are telling 1 gram or 0.1 to 1, 1 to 1. What is that? That is gram percentage, that is concentration or amount of sugar. So by seeing the color of the solution, you can calculate how much gram percentage of sugar present in the urine. That's why it is called semi-quantitative, not fully quantitative. If you will go for fully quantitative, that will be different tests are there. GUD, PUD, glucose oxidase peroxidase, orthotoluidine method, Nelson-Somogyi method, so many methods are. Okay, hexokinase method. Okay, but this is qualitative, and Benedict test also called as semi-quantitative. Why? Because by seeing the color of the solution, we can assume how much gram percentage of sugar present in the urine. Understand? What is the composition of Benedict reagent? Yes, copper sulfate, sodium citrate and sodium carbonate. Okay, 3 compositions are there. 3 have different roles. Is it? 3 components have different roles. What is the role of sodium carbonate? Sodium carbonate is there. What is their role? Sodium carbonate role is, it is a alkali. So makes the medium alkali. So now you can tell Benedict test. Which medium we are doing Benedict test? Alkali medium. Understand? Due to sodium carbonate. Makes the medium alkaline. And next is copper sulfate. Copper sulfate what will do? It will give the more cupric ion. Cupric ion. Okay. In urine what is there? Sugar is there. Reducing sugar is there. So that sugar, what it will form? It will form unique enediol. You will form 1,2-enediol. Understand? 1,2-enediol. So that enediol is powerful reducing agent. Understand? Powerful reducing agent. Understand? So what it will do? Then cupric ion is there. That cupric converted to cuprous hydroxide. Okay. Then after that finally it will reduce to cuprous oxide. Cuprous oxide, Cu2O. Cuprous oxide finally it will reduce. Okay. When we will go for Benedict test, okay, after that you will see precipitate part. Brick red, what is that? That is precipitated. Green color, that is precipitated. So that precipitated part, what is that compound? That is nothing, that is the cuprous oxide. Understand? That is the cuprous oxide. What is precipitated? That is cuprous oxide. Okay, Benedict test. And one thing is there, suppose false positive also may come. Benedict test. If it is patient is not diabetic, but Benedict test is coming positive. Why? Due to some reason is there. What is that? Suppose vitamin C, ascorbic acid. Suppose today you took vitamin C. Tomorrow urine will come Benedict positive. But it is not diabetic. Understand? Vitamin C, creatinine, urates, okay, they will interfere and they can be reduced. Understand? So this is your regarding Benedict test. Next is your heat coagulation test. Test for protein, heat coagulation test. How will do? You will take 2/3 part of the test tube filled with urine, then you will heat the upper part. Solution you will take up to this. Okay, 2/3 part. Okay, then you will heat just upper part. You will not heat up lower part. Just heat upper part. Why you will heat upper part? Why will heat upper part? Why only heating upper part? Because lower part you will take as a control part. lower part will take as a control, upper part is a test part. Understand? Test part, control. When you will heat upper part, only due to heating, what will that protein will be denatured here. Okay. First it will come turbid like it will come. Okay. Then you will add glacial acetic acid, 2 drops. 2 drops glacial acetic acid you add. What will see? You will see clear coagulum. Clear coagulum. Coagulum will see. That is protein will denature. How many structures of protein are there? Primary, secondary, tertiary, quaternary. Now due to denaturation, which all structure will denature or any structure will? Very good, very good. Primary structure will intact, it will not denature. Secondary, tertiary, quaternary will denature. Understand? Now you are adding glacial acetic acid. Why you will add glacial acetic acid? What is the role? Glacial acetic acid 2 drops you will add after heating. Why? Because what is the cause? Sulfates. These are present in sulfates, phosphates present in urine. It may interfere with coagulum. Okay. So when you will add the glacial acetic acid, sulfates and phosphates will dissolve. So clear coagulum it can be seen. Understand? Urine থেকে হ্যাঁ ইউরিনতে কি থাকে ফসফেটস থাকে and also sulfates থাকে, so that sulfates and phosphates will dissolve by adding glacial acetic acid. Okay, ফসফেটটাকে সালফেটটাকে dissolve করে দিলে clear coagulum, the protein denature, that clear coagulum you can find. Okay, understand? Next test is sulfosalicylic acid test. You will take 3 ml of urine solution, then you will add drop by drop 20% sulfosalicylic acid. Then you will see white precipitate is there. White precipitate, that means presence of protein is there. That whites like that in Bendick test, that precipitated part, what is that name? In Bendick test, that precipitated part, that is cuprous oxide. Same like in sulfosalicylic acid test, that precipitated, white precipitated will form. What is that name? That is protein sulfosalicylate. Because protein is present in the urine, adding sulfosalicylic acid, so it will form protein sulfosalicylate. Understand?
বলছিলাম যে যদি ওখানে আমরা মানে কিউপ্রাস অক্সাইডটা যখন মানে প্রেজেন্ট আছে কিউপ্রাস সালফেট ওটা তো কিউপ্রাস আয়ন দিচ্ছে
কিউপ্রিক দিচ্ছে হ্যাঁ
আমরা যখন এই অ্যাসিটিক গ্লাইসিল অ্যাসিটিক অ্যাসিডটাকে অ্যাড
গ্লাসিয়াল অ্যাসিড এদিকে গ্লাসিয়াল অ্যাসিড তো ওটা তো ইয়েতে তোমার ওটা প্রোটিন জন্য এটা হ্যাঁ ওটা হিট কোয়াগুলেশন জন্য এটা নয়
এটা ফর্ম করছে হচ্ছে কিউপ্রাস অক্সাইডস আর
এটা কিউপ্রাস অক্সাইড ওটা হচ্ছে প্রোটিন সালফোসালিসিলেট এসএসএ ঠিক আছে এবার দেখিয়ে দিচ্ছি দেখে নাও সো দিস ইজ 15 ml so up to this you can take that is 5 ml okay हाँ अरे
স্যার 8 ড্রপস
ये तो सही है
আচ্ছা উপর থেকে
जो पहले आया वो आजा 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 तो नीचे वेट कर रहे हैं
একটু সাইড
test part. Understand? Upper part you will heat why? Lower part you will take as a control. Like that you will, when you will do for bile salt, what you will do? Test another control. For control what you will do? Only distilled water you will take distilled water, then pinch of sulfur powder will sprinkle. Understand? If positive is there, then sulfur powder will sink down. If negative is there, sulfur powder will just flows. Understand? Like that. This is also here also. This is your control part. Test part. Keep it. The.
It's like a 3 ml urine.
3 ml urine solution. Then what you will do? Sulfosalicylic acid. How much you will add? Drop by drop. Okay. Presence of protein, white precipitate. This compound is, what is the compound? Compound is the protein sulfosalicylate.
ফ্লেমটা থেকে সব ফুটে যাচ্ছে।
আমি জানি স্যালাডের মধ্যে কোনো ইয়ে
অ্যাসিডিক অ্যাসিড
এটাকে ট্যাবলেট না প্রোটিন নিয়েছি
না না ঠিক আছে
এই পুরো এই কন্টিনিউয়াসলি করিস না ওটা
ওই
আর নানাবি না।
সুচরিতা ওই ইউরিনটা যেতে এদিক দিয়ে
এই তিউরিন
আরে দেখো এখানে
এটা ঢেলে নেই, এভাবে ড্রপারে হয় না, ঢেলে নেই। সুচরিতা হোল্ডারটা দে, হোল্ডারটা
বেনিফিটস তো সহজ আর কিছু
কাল লাগবে
কোনটা দেন এর কি উপরে একটা
মানে আরো
এটা তো আমাকে হোল্ডারটা দে।
দেখি শেষ হয়ে যাচ্ছে
ये